Hello, this biomedical instrumentation lecture will cover microelectrodes in cortical neuroprosthetics. Previously, we have been concerned with measuring potentials on the body's surface, such as EKGs and EEGs. In this lecture, we're going to talk about electrodes that are capable of making single-cell potential recordings. That is, we're going to be talking about invasive measurements. So microelectrodes are devices that measure potential differences across cell membranes. They are small with respect to cell dimensions, and they are typically made to be strong so as to remain mechanically stable. They also have tip diameters that range anywhere from 0.05 microns to 10 microns. Let's draw a micropipette or glass capillary electrode. So this type of electrode will typically have a tip diameter of around 1 micron. And let's first draw a cell that it would be poked into. So we have some potential difference across the cell membrane. Represented by opposing positive and negative charges on either side of the cell membrane. I'll draw a nucleus. And then I'll draw the electrode itself. So this is typically going to be tapered at the tip. Probably drawing the tip a little bit too big there. And it's going to have, looking through it, it's going to have these glass walls. And then it'll have a cap on the end through which a metal electrode will be inserted. Which will lead to a wire coming out of it. We'll call that V-lead. You'll also have to have some kind of reference point, some kind of grounding reference point, in order to measure a potential difference between the electrode itself and the rest of the body. Here's the tip. This is the inside of the cell. The micropipette's impedance turns out to be mostly resistive, with a resistance that's somewhere between 1 megaohm and 100 megaohms. And here's an equivalent circuit for a micropipette electrode inserted into a cell. You have this voltage source, which is time varying. You have a resistance, a capacitance. You also have a half cell potential. So we'll call this capacitance C sub t, the resistance R sub t, and the half cell potential E sub m. And here, the voltage source represents the membrane and action potential. The sum of those two, which could be time varying. That's what we're interested in measuring. Okay, so R sub t here represents the overall series resistance of the electrolyte. And C sub t represents the total distributed capacitance, which I believe is dominated by the glass, because the glass itself acts as a dielectric. The E sub m are DC potentials at electrolyte and metal electrolyte interfaces. They result from unequal distributions of charges, non-uniform distribution of charge at those interfaces. Now you probably recognize this sort of circuit from previously in our class, where we had a voltage source connected to a resistor, and then you read out the voltage across a capacitor. That looks like a low-pass filter. So with that parallel capacitance, the micropipette has a low-pass filtering effect on the source potential that you're measuring. So we have to counteract that using positive feedback, or equivalently said, negative capacitance amplifiers that effectively cancel out that capacitance. But mostly, overall, it's resistive. Okay, so microelectrodes, micropipette electrodes, are an old technology with many drawbacks. For example, it's hard to make a lot of them. You have a low manufacturing volume. So it's hard to make many in a repeatable manner. There's also great injury potential to the cell that you're trying to measure, and it's relatively invasive overall. Furthermore, in many, if not most, clinical applications, recording from a population of cells, not just one cell, is necessary. And we want chronic recordings over years. We want to be able to embed a device once and leave it there and know that we're going to have good measurements for years to come. So in the 1980s, researchers started to translate semiconductor techniques to microelectrode construction, yielding what are called silicon micromachined microelectrodes. Silicon micromachined, I'm going to have to write in micro handwriting to fit it all on here. Silicon micromachined microelectrodes. And one of the first ones was the Michigan probe. So this is a semiconductor device that has it's a semiconductor device that has multiple measurement sites on it. And later in class we'll talk about how these kinds of devices are created. But what I want you to appreciate here is how it has these multiple gold recording sites, right? You have one there, one there, one there, one there. And these each have their own lead coming off that you can then connect to subsequent amplification circuitry. And this probe really broke a lot of ground, and in the early days of measuring in vivo neuronal, making in vivo neuronal measurements, and it's been used extensively in research to develop cortical neural prosthetics. Or CNPs. So the advantages of silicon micromachined microelectrodes over micropipettes include the ability to do very precise construction. You get both precise construction electrically, that every probe you make has the same electrical properties, and geometrically. You also have now the ability to build arrays of probes with very precise geometries. Typically, one probe that we're going to insert in the brain is not going to cut it. We need to have lots of different measurement sites. They cause less injury to cells than micropipette electrodes, and it's once you've built one, once you've set up the machinery to build one of them, building more is very cheap. So it's cheaper to build them in large quantities. They do have some challenges though. One is in installation, in that when you open up the brain and insert them, you get swelling or shifting of the probes after you've inserted them. You also get a strong immune response to insertion. And so what that manifests as is a sheath of cells that can wrap around the electrode and isolate it electrically from the neurons. So a lot of research now goes into figuring out what we can coat these probes with to trick the nervous system into not generating this immune response. You also have larger tips than with micropipette electrodes. So it's not really possible with one of these to make isolated single cell recordings. Another problem is that the shank gives a large series resistance on the order of 10 to 100 megaohms. So for patients without the ability to move one or more of their limbs, cortical neural prosthetics hold significant promise to restore motor abilities. So ideally, we'd like to have this kind of a setup. We'd like to have the body here. We'd like to be able to embed some probe in the brain, do some processing on the signals that we get out of it, and then send some command signals to another related type of probe that's embedded in muscle. So ideally we could control muscles directly using activity measured in the brain, or in the motor cortex specifically. This essentially skips or bypasses most of the central nervous system. But currently, due to difficulty in controlling muscles externally, research has mostly focused on how we can decode signals from neurons in the motor cortex to infer what the subject wanted to do, and then use those signals to control something like a robotic arm or a computer cursor. So this is sort of the setup we have now. We can measure the signals, we do the processing, and then we infer some commands that we send to like a robot arm to move. Count my fingers here. And so where do we get the signal from? We get it from the motor cortex, right? And the motor cortex, as we know, is organized topographically, much like the sensory cortex. And so by inserting a probe in a given location that represents the region of the body that we want to control, we can put a probe here and try to figure out, okay, what is it that the subject wants to do with their arm, right? And this map is called a homunculus, which is one of my favorite words. But there's a problem here. We have this topographic mapping, so we can generally get within, a centimeter or so of what body region we're interested in. But it's nearly impossible to find one single neuron within that motor cortex that encodes a given muscle contraction or motion. Furthermore, neuronal firing is a statistical process. All neurons fire all the time. It's just the rates that change. And control signals are so based on that, control signals are derived from the rate of a population of neurons. Sorry, from the firing rate. Of a population of neurons. So how do we go from population firing to muscle movement? There are various approaches. And there's two basic approaches here. One are model-based approaches. So what you do there is you calibrate a model that relates firing rates to rotations, limb rotations. This is an inferential approach. The other approach is essentially machine learning based. And in this case, we say we don't know what the mathematical relationship is between the firing and the intended rotation. So instead, we're going to build a dictionary using automated methods and training data, where we make a bunch of measurements while we ask somebody to try to do a certain thing. And then we compare each measurement to each entry in the dictionary, and the most similar entry wins and must be what the user intended. This is a model-free approach. We essentially use machine learning to implicitly learn a model without having to define it explicitly ourselves. However, it is now believed that the actual method or algorithm that is used to convert firing rates to motion is not as important as establishing a closed-loop system. That is, we let the body tune its own outputs based on its own observations of what those outputs did. So as long as you have the subject able to view the results of what's happening based on their brain activity, that's all that really matters. And the brain is plastic enough that it can figure out how to generate the right signals to do what it wants. So in this schematic, we have this feedback loop. We have the brain, goes to the brain-computer interface, goes to the robotic arm, and then the eyes observe what the robotic arm is doing and represent the feedback to the brain to adjust its output. And of course, in addition to brain-computer interfaces, there are lots of other microelectrode applications, chief among which is probably cochlear implants. But there are also retinal implants, stimulators to relieve pain in the spine, stimulators also for bladder control, and deep brain stimulation. And then there are also emerging hippocampal stimulation devices to restore long-term memory. In this case, what we have is a cognitive prosthetic. So that's our lecture on microelectrodes in cortical neuroprosthetics. I'd invite you to go visit the Human Neural Prosthetics program at University of Pittsburgh to see some really cool videos of implanted electrodes in humans, and then using these to control different devices. The Schwartz Motor Lab also has a lot of great videos showing both human and non-human primate experiments where they're able to control robotic arms and things just by thinking about it. And the interesting thing when you're looking at these is that you notice that originally when they were training the non-human primates, they were always trying to move the limb that corresponded to where they were taking the measurements from in the brain. But eventually the brain rewired itself, and you ended up seeing no motion in the limb. And so it was literally the primate was able to actually essentially grow a third arm, which is pretty amazing when you think about it. Okay, that's it. Thank you.